A1 Gene Expression Summary for Human Biology Level 3 BTEC. So gene expression is a process by which a gene produces its product and the product carries out its function. So usually the product will be a protein. So when a gene produces a protein and then that protein carries out its function. Transcription is the process of making a complementary copy of one of the DNA strands in order to make a complementary strand of messenger RNA or mRNA. And a gene is a section of DNA on a chromosome that codes for a polypeptide or protein. So the steps in transcription. The first thing is that the hydrogen bonds between DNA bases break. Only one DNA strand acts as a template. 3 RNA nucleotides align by complementary base pairing. In RNA, uracil base pairs with adenine. RNA polymerase joins adjacent RNA nucleotides by phosphodiester bonds, and then the pre-mRNA is spliced to form mRNA in order to remove the introns. Some definitions now. So a codon is a specific sequence of 3 adjacent bases on a strand of RNA that provides genetic code information for a particular amino acid. So the codon is a sequence of 3 on the mRNA. An anticodon is a group of 3 bases on a tRNA molecule that are complementary to an mRNA codon. And transfer RNA is a type of RNA molecule that transfers amino acids to ribosomes during protein synthesis. Amino acid activation is the process of loading an amino acid to the tRNA using ATP. So now looking at translation, which is a process by which mRNA is decoded and a polypeptide is produced. So the steps of translation. First thing is that the mRNA attaches to ribosomes. The tRNA anticodons bind to complementary mRNA codons, and tRNA brings a specific amino acid, and then the amino acids join by peptide bonds, and this requires ATP. Then after that, the tRNA is released after the amino acid is joined to the polypeptide, and the ribosomes move along the mRNA to form the polypeptide. Now looking at mutations. So gene mutation is a change in the base sequence of a gene. And gene mutations usually occur during DNA replication. So there's 3 types of mutations. You've got deletion mutation, which is where one or more pairs of nucleotides are removed from a gene. You've got a substitution mutation, where a single base is replaced, potentially altering the gene product. Insertion mutation, one or more nucleotides are added to a gene. So deletion is taking one away, substitution is swapping one, and insertion is adding a base. How a mutation can result in a non-functional protein. So the primary structure of a protein would alter if the amino acid is changed, which could change how the protein folds in the tertiary structure. Obviously, if that's an enzyme, that's quite important because it could mean that the active site is a different shape and therefore won't bind to the substrate. If there's a mutation in the stop codon, this results in a shorter polypeptide chain being produced. Now, looking at germline mutations, these are DNA alterations which occur in the gametes that can be transmitted to offspring. And then somatic mutation is a mutation that occurs in the body cells, and this cannot be inherited.